Good morning, students. Today's class we are discussing about the economic importance of fungi. Last session we have seen all about bacteria, its economic importance, its structure, everything we have learned. So before we are dealing with the class, let me know what is meant by fungus. The word fungus you are familiar. Do you know where you can see this fungus? Yes, the common thing that it is a bread mold. You can see if a bread piece is kept outside for 2 to 3 days, you can see a blackish or greenish or grayish structure which is growing on this bread, which you call it as bread mold, which is a fungus. Then come on, say another example for a fungus. Yes, you can see that in the lemon. If you cut a piece of lemon and keep it outside, you can see that there also a blackish structure which is growing, that is nothing but fungus. So again on leather items also you can see, especially in rainy season, if in the presence of little moisture and warmth, you can see this fungus is growing. So everywhere we can see this growth of fungus. So you can say that first we can study about the bread mold which is called as rhizopus. So bread mold which is growing on the bread, that is called as, its name is rhizopus. Coming to the structure, if you are keeping that structure under a microscope, you can see fine hair-like structure, and these fine hair-like structures are called as hyphae. It is nothing but hyphae. So these are the fine hair-like structures which is called as hyphae. And these hyphae together it is called as what? Mycelium. So the fine thread-like structure together it is called as mycelium. The two terms, that is, one is hyphae and the collection of this hyphae is called as mycelium. And yeah, then you can see that this hyphae, it will produce at the time of nutrition, or if it wants to consume the food content, usually just think that it is in the bread is the substratum. And so in the bread, you can see that it will just produce an enzyme. Suppose this is the bread piece, you can see that on the bread it will just produce an enzyme, and in this enzyme its food material will be absorbing, and the nutrient it will be absorbing. So and the absorption takes place from outside itself. So it is called as extracellular digestion, because it happens, the digestion happens outside the cell, so we are calling it as extracellular digestion. So intracellular digestion usually in case of our body and all inside the cell, so it is intracellular digestion. So that quite opposite meaning. Extracellular means outside the cell digestion takes place, that is called as extracellular digestion. And coming to the reproduction of these fungus, especially bread mold or rhizopus, you can say that at the time of reproduction, few hyphae, this hyphae, it will become erect in structure like this. So here and there, you can see this hyphae, it will become erect structure or a straight structure like this. And at the end region, you can see it becomes swollen. At the end of this erect region, it will become swollen, and that structure is called as sporangium. So this erect structure, and at the tip sporangium is formed. And what you can see inside the sporangium? Tiny spore-like structures. So tiny spore-like structures you can see, and these spores on maturation, you can see the sporangium when it gets matured, these spores will be, this sporangium breaks open and the spores will be liberated outside. And when it reaches a suitable another substratum, maybe a bread or any other substratum, when it reaches there, it started to start a new life. That's how reproduction takes place, and this is one of the essential method of reproduction that is usually seen in fungi. So you have seen where these fungus are seen almost everywhere. You can see even on the woods, you can see, or on the bark of the trees, or fruits, or leather items you can see, food materials it will grow, in bread it will grow. So everywhere it is common, and its structure is also quite simple. And so rhizopus, you can see, this is the structure, how the bread mold is forming. And whether this fungus has any nucleus, children? Yes, this fungus has well-defined nucleus with nuclear membrane. So it comes under the group eukaryotes. So prokaryotes, while bacteria you are studying that only nuclear material is present, whereas specific nucleus with the nuclear membrane is absent. Here you can see that a definite nucleus is present in this fungus. All the fungi you can see the nucleus. So it comes under the group eukaryotes. And can you say whether any this chlorophyll is present in this one? Yes, it is no. There is no chlorophyll present because it is non-green in color, and so automatically the mode of nutrition will be whether it is autotrophic or heterotrophic. Yes, the mode of nutrition is heterotrophic. So the mode of nutrition is heterotrophic, and I told you how this get its food by just releasing an enzyme on the substrate and which food material it is growing. There it is releasing the enzyme, and just it is extracellular digestion takes place in this one. And again at the time of reproduction, you can see a erect structure and a straight structure will be forming, and at the end of this region, this swollen structure formed which is called as sporangium, and the sporangium breaks open, and what will be coming outside? This spores, and these spores when it reaches a suitable substratum just like bread or something, it starts to grow there. That's all about this rhizopus, which is a bread mold. Next, another fungus which is common is yeast. So you may be familiar with the word yeast. Usually in bakeries and all, you can see that for making bread, bun, etc. this yeast is used. It's a unicellular structure. It is bit oval in shape. So what is the structure? You can see the shape is oval. It is oval in shape, and it is covered by a cell wall. The outer membrane that is called as cell wall. So the outer membrane is called as cell wall. Inside the cell wall you can see one or two or three. That is more than one vacuole is present. Some big vacuole is there. Some other small vacuoles are present, and again some granules small like this. Granules are also present in this yeast cell. Then again a well-defined nucleus. Here you can see a nucleus with a nuclear membrane is present. So a nucleus with a nuclear membrane is present in the case of yeast. That's the structure of yeast, and let us see how it has taken its nutrition. It's a simple way that you can see that the sugar, it is present in the cane sugar or sugar. You can see it is broken down into simple sugar, and it will be absorbed into the plant body. So that is a simple way of nutrition. And coming to the respiration, what is the type of respiration usually happens in the case of this fungi? Already in the last session I have told you that some bacteria as well as fungi usually they respire by means of anaerobic respiration, that is in the absence of air only these will respire. And can you say the equation for anaerobic respiration? Yes, glucose method of respiration we can see in this fungi, that is anaerobic respiration. And what is its importance to mankind? Yeast have great importance to mankind, mainly in the case of alcoholic fermentation. You can see this alcoholic fermentation. In order to get wine or beer, whiskey, gin, etc. All these cases we are using this yeast. So it has a great economic importance since ancient time onwards. Now let us see few useful fungi. Already in the last session I have told while I am taking about bacteria and its uses. I told about a first antibiotic. Who discovered that one and what's its name? Can you recollect that one? Yes, the first antibiotic is nothing but penicillin. It is actually a fungus. It is from a fungus. It was a milestone to this, or we can say it is a wonder drug in the field of medicine. That is the first antibiotic, which is Penicillium notatum. Its scientific name is Penicillium notatum. It is a fungus. From that, what we are extracting the antibiotic, which is called as penicillin. It is very useful. It's a very useful drug or medicine. And now harmful fungi, we can see that there are two fungus, especially Aspergillus and Penicillium. Don't confuse with the penicillin. Aspergillus and Penicillium. This is called as blue and green fungus respectively. This Aspergillus as well as Penicillium, green and blue fungus, and this will you can see which grows on the outer covering of this lemon, orange, etc. Sometimes when you are buying an orange, suppose the outer, usually that orange color, a slight bluish or greenish color comes, means there is no doubt that is the growth of the fungus. It is nothing but the growth of fungus. Clearly you can observe. And useful role in the fungus, I already told you in the production of antibiotic. What is meant by antibiotic? Already you have seen in the previous classes that it's a chemical produced by a living microorganism which can stop the growth of certain other harmful bacteria. And what are the features or characteristics of a good antibiotic? Also you have seen that it can be used for more than one infection. The antibiotic should be used. It should not have any side effects and it should not kill other bacteria which is living in the body. These are the characteristics of a good antibiotic. And what are the roles of this fungi in industry? Now we can see what are the roles of this fungi in industry. First one you can see, already I told you, it is used in the production of wine. So in the case of wine, by the process called fermentation, a process named fermentation, we are getting the substance wine. Now what is meant by fermentation? So, what's the meaning of fermentation? The process by which microorganisms just like yeast or bacteria breaking down this glucose or carbohydrate into simpler substance like alcohol or ethyl alcohol or lactic acid. This process is called as what? Yes, fermentation. This process is called as fermentation. Once more, the process by which microorganisms like yeast or bacteria, they break down this simple substance like carbohydrate and sugar into ethyl alcohol or lactic acid, which is called as what? Fermentation. And in the production of wine or in the production of, you can see, alcoholic substances, we are using this process fermentation. This happens in the presence of yeast, which is a fungi. So already you know the equation of anaerobic respiration, that C6H12O6, that gives what is the substance? So C2H5OH, that is nothing but ethyl alcohol by liberating which gas? Carbon dioxide gas. So carbon dioxide is released during this process. Now, how this wine is prepared, let us see. Usually, you know, wine is prepared from which substance? What is the source of preparation of wine? Yes, that is grapes. So, this grapes is crushed. So, breaking down into small pieces, that is crushing process is done, and then it is fermented in a large vessel. So, grapes are crushed and then it is fermented. How it will be fermenting? Yes, already these yeast cells will be present in the grapes. Apart from that, we are adding what? Yeast, so that the fermentation process will be takes place. So you can see that in presence of this yeast in the crushed grapes, what will happen? Fermentation takes place and a substance called ethyl alcohol will be produced. This ethyl alcohol, when it reaches at 12%, when its concentration reaches 12%, what it will do? This ethyl alcohol will kill the yeast cells, and what is left behind is nothing but wine. Understood? The process of preparation of wine. Once more, these grapes are crushed and fermented in a large vessel. So, how the process of fermentation? By adding yeast cell. Already yeast cells are present in grapes. Even then, we are adding some more this yeast cell so that fermentation takes place. At the stage that is about 12 degree, this when it reaches a 12% concentration, this wine, when it reaches 12% concentration, what it will do? It will automatically kill the yeast cell, and what is left behind is nothing but wine. So from this, again more concentrated whiskey or gin we can prepare. How this process will be taking place? These wines we have to take, and then it has to undergo distillation. What is meant by distillation? In the previous classes you might have studied about distillation, especially in chemistry. What is the process of distillation? Yes, distillation is a process by which heating the thing. Say for example, distilled water. You are getting distilled water. How it is doing? The ordinary water we are heating and allow the vapors to collect in another chamber. By cooling these vapors, we will get again water drops, that is called as distilled water. So same procedure only here. The wine we are distilling, heating it. From the wine, the pure vapors will be coming out. The vapors will be collected through a glass tube and or big pipes. In industry, it is not through a small pipe, through a big pipe, and that will be cooled. On cooling outside the pipe, water will be poured, and when we are pouring this water, this gas or vapor, that will be changing into liquid. This is the process called the distillation. After this distillation, we are getting this whiskey or gin which have more alcoholic concentration. So the preparation of wine as well as the preparation of whiskey you understood. One by ordinary by wine by fermentation using yeast, and whiskey by distillating this wine. Now what are the characteristics of this wine? Yes, wine is quickly absorbed by the body. When we are consuming this wine, that will be very quickly absorbed by the body. And in mild quantities, that means if you are consuming in small quantities, you can see it is a stimulant. It will stimulate our appetite and all. But if we consume in large quantities, what will happen? You can see that it is harmful in many ways. So be careful that in a small quantity it may act as a stimulant, but if you consume this wine in large quantity, it will have some harmful effects on the body. What is this? It may develop body ailments, some diseases in the body, and particularly in the liver cirrhosis. So liver cirrhosis is it will damage the liver. It's a condition that the cells of the liver is start to get damaged. Actually, liver is called the chemical factory of our body, because all this synthesis, you can see even the poisonous substances, the liver only will be removing through our blood. So here liver is called the chemical factory, and if you consume even alcohol also consuming in a large quantity, first it will it will destroy the liver. So these are the characteristics of wine. Once more, it is quickly absorbed by very fastly it can be absorbed by our body. In mild quantity or in small quantity, we can say that it may be a stimulant to our body, whereas if you consume this wine in large quantity, it will have some harmful effect. Say for example, if you are consuming this wine in large quantity, your liver may get damaged, leading to liver cirrhosis. Now another important, so how wine is prepared we have seen and its characteristic. Now in bakery how this yeast is used, let us see. So in baking, you can see in bread making, say for example bread bun, all these cases we are using what? Yes, this yeast. So first let us see this bread making, how this bread making is done with the help of this yeast. In bakery, the mixture of flour, which one? The maida, the flour, wheat or maida we are taking. It is mixed with little fat, and then salt and water we are adding. Then we are adding little sugar, and along with what? Yeast we are adding. So we are stirring it well and we are making it a dough. And in the dough, you can see the same process, anaerobic respiration of yeast takes place, and what will be producing the gas that comes out is nothing but carbon dioxide. The dough we are making after mixing this salt, water, fat and the flour, and again we are adding yeast cell. The fermentation process takes place, releasing carbon dioxide gas. This carbon dioxide, when escape from this dough, you can see that it leaves some openings, because this gas wants to escape from this dough. So when we are baking, you can see that 1/3 volume, the size will be increasing. A small amount of dough we are keeping for baking, you can see that third time its volume will be increasing, and you are getting very spongy bread. And how it becomes spongy? Because these air carbon dioxide gases, first thing I told you, when it is leaving out of this bread, it makes this bread spongy, soft and spongy, and thus it becomes aerated. Thus it happens. So the production of bread or making bread, you understood. Now how the production of cheese used, let us see. It's a valuable food. This is also one of the product of this yeast cell, and it is a valuable food with the high quantities of proteins, fat along with calcium and phosphorus. So you can see that in the cheese, you are familiar with the cheese, which is one of the milk products. And what are the elements present, or what are the nutrients present in this cheese? Yes, you can see that protein is present, fat is present, then calcium, phosphorus and vitamin A. These are the nutrients present in this cheese. And from centuries you can see that means in ancient time people were using this cheese. And how it is made? By the 4 steps are there for making this cheese. First step is curdling of the milk. So that is nothing but milk changing into curd. By the how we can do this one? By the addition of lactobacillus. It's a type of bacteria that is converting this milk into curd. So first thing is that milk changing into curd by the addition of lactobacillus or lactic acid. So the first step is called as curdling of milk. Second, afterwards you can see while milk changing into curd, a watery liquid will be present in that one. That you have to separate. And this watery liquid, what is present is nothing but we call it as whey. One of the byproduct of curdling of milk, it is called as whey. And then second step, processing of this curd. How this processing is done? If still any water is there, we have to remove that water and moisture from the curd. That process is called as this processing of curd, and that time we are calling it as what? Cottage cheese. That time what the product we are getting, that product we are calling it as cottage cheese. So first one is curdling of milk by the addition of lactobacillus. We are changing this or converting this milk into curd, and the watery liquid what it is present, that is called as whey, that is removing from this curd. And now this processing this curd, how it is done? By removing the moisture content from the curd. Third process is called as salting. From the name itself, you know, it is the addition of salt. We are adding what? Salt. So why we are adding this salt? To remove the moisture. Still any more moisture is present, we have to remove that moisture. For that we are adding the salt. So the third process is called as salting, and one of its purpose is to remove the moisture content from this curd. And again another thing is also there, that is that if any undesirable microorganism is present in that cottage cheese, that also has to be died. So the salting is done in the third stage. And fourth stage is called as ripening. In that the curd is kept in a suitable temperature and humidity. So in a particular temperature and humidity, the curd is kept, and again the addition of microorganism here also. Which is the microorganism? Yes, it is lactobacillus, a type of bacteria which is working on this one, and this add a particular flavor to the cheese. So the production of cheese is over. It usually happens in four steps. One is curdling of the milk, that is milk changing into curd by the addition of lactobacillus, and the watery liquid we are removing. That liquid is called as whey. We are removing that one, and during the second process, that is processing curd, that is done by removing the moisture content as far as possible. And that time after removing this moisture content, we are calling this curd as cottage cheese. During the third step, we are adding salt so that if still any more moisture is present, that can be removed by the addition of salt. And again, if any microorganism is present, that also can be removed by this addition of salt. During the fourth stage, it is called as ripening. During this time, the curd is kept in a suitable temperature and humidity, and still again the addition of microorganism, that is called as lactobacillus. You can say that it will get a particular flavor. That time, what the product we are getting, that is nothing but cheese. If you have any doubt, within 2 minutes you can send a message. If you have no doubt, we can proceed the class. So next about mushroom. You are familiar with the term mushroom. Mushroom is also another example of this fungi. And there are different types of mushroom available. You can see, especially in the rainy season in the soil, different kinds of mushrooms are coming up. Can you say that whether all these mushrooms are edible or not? What is meant by edible? Which can be consumed by human being. Can you eat all these types of mushrooms? No, we can't eat these mushrooms because only few mushrooms are edible, others are highly poisonous. Sometimes if we eat some poisonous mushroom, it may lead to death also. So don't try with all type of mushroom which is available in the soil. Only few mushrooms are edible. And this, what is the scientific name of this mushroom which is commonly edible? That is Agaricus bisporus. Agaricus bisporus. Agaricus is the genus name. Bisporus is the species name. So this mushroom, it is an edible one, and usually you can see this mushroom, it has a cap-like structure like this. Yes, and a stalk. And here you can see small gill-like lines like this, which is called as gills of the mushroom. So this is called as the cap of the mushroom, and this is called as the stalk of the mushroom. These are the two main parts. And here inside the cap, small layer-like structure is present, which is called as these gills of the mushroom. And when a mushroom matures, inside these gills, small structures are produced, very small structures, which is called as seeds or spores of this mushroom. So there are mainly three types of edible mushrooms are there, which we can consume. One is white button mushroom, which is commonly available in the market also. And next one is paddy straw mushroom. Second one is called as paddy straw mushroom, and third one is oyster mushroom. These are the three types of edible mushrooms which are commonly available. And this mushroom cultivation we can done in indoors. Usually you can see it will be available in the rainy season. From the soil it will be coming out within 2-3 days. You can see the decay also will take place. At home we can see that we can cultivate this mushroom. If you have a little space, definitely you can start cultivating this mushroom. And what are the steps including in this mushroom cultivation? First one is called as composting. So what is the purpose of composting? That we have to make ready a substratum. A place should be ready for this mushroom to grow. That is called as composting. And it is prepared by, how this compost is prepared? By mixing certain substances in certain proportion. And first one is paddy or wheat hay we are taking. What is meant by paddy hay? The leaves left behind after harvesting the grain from the paddy plant, that is called as hay of the paddy, plus some chicken manure. Then you have to add some fertilizers, mix it well. So the compost is ready. The first thing is composting. Composting is now ready. What are the things needed for that one? Paddy hay or wheat hay. Hay is nothing but the grass left after harvesting in the paddy plant. Okay, so that one you have to mix it with the chicken manure. The chicken waste you have to add. Again you have to add some inorganic fertilizers. So that mix it well and keep it for 50 degrees Celsius for 1 week. You have to keep this. As soon as mix ready, this after mixing this compost, immediately you don't do anything else. For 1 week you have to keep it for 50 degrees Celsius. Now composting is over. Second one is called as spawning. Spawning is nothing but sowing this mushroom seed in the compost. So the mushroom seeds that also available, usually you can see in agricultural farm and all, these mushroom seeds or you will be available, and even in that you can see that some nurseries also you have this mushroom seeds, and these mushroom seeds, just that you are sowing these mushroom seeds in the compost, that is called as spawning, and this is allowed to spread for a couple of days. Means for 2 days minimum you have to spread upon, suppose this is the substratum, upon this you have to sow the seeds and keep it like this. Next, third step is called as casing. So third step is called as casing. During casing, what we are doing, a thin layer of soil we are spreading upon this mushroom seeds. So here mushroom seeds are there. Upon that we are spreading a thin layer of soil. What's the purpose of adding this thin layer of soil? Yes, you can see that it gives support to the mushroom. Soil gives support to the mushroom, and again it provides humidity. How much water is needed, the soil will provide, and prevent quick drying of the compost and helps to regulate the temperature. These are the four important things the soil does in the case of this mushroom cultivation. First one is it provides support to the mushroom, then it provides humidity, and third case it prevents quick drying of this soil and or the seeds, and it helps in regulating the temperature. Next, fourth stage is called as cropping and harvesting. Cropping and harvesting is from the name itself you know that when the mushroom you can see then a pin, just like a pin's head. This is a substratum, very small point like a common pin you know. It has a head region, very small it is in nature. Just like that it has come out. So during cropping and harvesting, first it appears as small pinhead like region, then the hyphae and mycelium spread on this compost, and then you can see slowly the button like structure will come out, that is nothing but this cap of this mushroom, and clearly you can see the stalk also will be visible, and it takes 2 to 6 weeks. So you don't think that immediately after spawning or the seed sowing you don't get on the next day or the very next day these will not come out. Nearly 1.5 months is needed, that is 2 to 6 weeks time is needed for this mushroom to come up. Then it grow bigger in size, and when it achieves the size, maximum size of the fully grown mushroom can be cultivated. That is about cropping and harvesting, and the fifth stage is called as preservation. So last stage, this is the last stage. This is called as preservation. You know, I already told you that these mushrooms are highly perishable. Its life within 2 days. Suppose it is in the soil, within 2 or 3 days it will decay. So it is highly perishable. So it is very important to preserve this mushroom for the future use. How it will be doing? You can see that first one by vacuum cooling this in the absence of air. Cooling the mushroom in the absence of air, that is called as vacuum. And second one by passing gamma radiation, so that we can preserve it for few days, and storing at 15 degrees Celsius. So these are some of the method, and again by freezing also we can preserve this mushroom. So these are some of the methods of this mushroom cultivation. First one is composting. Next one is called composting, means we are preparing this compost by mixing what are the things? Yes, this paddy hay, then chicken manure and adding some inorganic fertilizers. Here the compost is ready and keeping it for 50 degrees Celsius for a nearly, you can see, 1 week we are keeping. And next is called as spawning, that is we are sowing the seeds of the mushroom, which is called as post. You don't think that by listening the word seeds, it is not so bigger in size, it's very minute, small spore-like structures, and that is called as the spawning. And third stage is called as casing, that a layer of soil we are adding to this compost. Next, you can see the cropping and harvesting. From that, the hyphae, mycelium, etc. will be growing. Then a small pinpoint like structure growing, small, slowly it will grow, and then you can see that the fully grown mushroom will be coming out. And then after harvesting this mushroom, the final stage is preservation. You have to preserve this for the future use as it is highly perishable. That's all about this class about the economical importance of fungus. Thank you.